Have you ever stopped to truly consider how your body knows what it feels?
Mm,
Think about like a cool breeze on your skin, or the texture of some fabric, or even that sudden sharp sting when you stub your toe.
yeah.
How does a physical touch or, a change in temperature transform into a clear sensation your brain actually
It's
understands?
an astonishing feat of biological engineering, isn't it?
Mm,
We navigate our entire world through these sensations, yet the underlying system is so incredibly precise and complex. It's just operating silently in the background.
absolutely. Welcome to the Deep Dive, where we take complex topics and really explore them, aiming for those profound, fascinating insights.
Hmm.
Today, we're embarking on a pretty ambitious deep dive into the somatosensory system.
Right.
Our mission for this session is to truly understand this incredible system, like the most knowledgeable experts in the field. We're going to unpack exactly how your body gathers information about its own state and its contact with the world.
Turning simple mechanical touches or thermal energy into electrical signals the brain can interpret. It's fundamental.
We'll trace these signals right from the start, from the initial sensory receptors, those tiny biological detectives, you could say, all the way through these intricate neural pathways.
Right up to the processing centers in your brain.
Yeah.
Yeah. We'll explore everything from the subtle nuances of touch to the, well, profound complexities of pain.
And even how your brain actively regulates what you feel.
Exactly. Our guide for this journey is a comprehensive look at the latest physiological research, drawing extensively from BERNE and LEVY physiology. So solid grounding there.
Okay, excellent. Let's start at the very beginning then, with the foundational layer. What exactly is the core function of this somatosensory system? What's its main job?
At its heart, its job is providing your central nervous system, your brain and spinal cord, with crucial information.
Okay.
Tells your brain about the state of your body itself, like, where your limbs are in space, and critically, how it's interacting with the outside world.
Huh.
And it does this by converting various forms of energy, could be mechanical pressure, stretch, vibrations, or thermal energy, into electrical signals.
Yeah.
That's the key conversion.
Right, transduction. And these initial electrical signals generated right at the sensory receptors.
Yeah.
Those the generator receptor potentials, yeah.
Precisely.
Yeah.
And they then trigger this cascade of action potentials, essentially electrical impulses that carry the specific characteristics of that stimulus, things like its intensity or duration.
Okay.
The journey begins with what we call first-order somatosensory neurons. Their cell bodies, the neuron's core, are mostly located in clusters called dorsal root ganglia for the body,
Mm-hmm.
and in cranial nerve ganglia for your head and face.
And these cells have a unique structure, right? Pseudo and polar.
That's right. It's fascinating. Imagine a single short extension from the cell body that then just splits into two branches.
Okay.
One branch reaches out to the periphery, acting as the functional receptor. Sometimes it's even encapsulated by specialized accessory cells.
Mm-hmm.
And the other branch heads directly into your central nervous system. It's almost like a dedicated hotline for that specific sensation.
So that CNS branch enters either the spinal cord through a dorsal root or the brainstem via a cranial nerve.
Yep.
And once inside, it branches out quite a bit, making connection synapses with various other cell types, including these second-order neurons.
Right, and these second-order neurons, at least the ones involved in conscious perception, then project up to specific nuclei within your thalamus. That's where the third-order neurons hang out.
And those third-order neurons are the final link in this sort of three-neuron chain for conscious sensation.
That's the main pathway, yes. They project to your primary somatosensory cortex, SI, we often call it, which is responsible for your conscious perception and more advanced processing.
Okay.
But it's really important to remember, somatosensory info isn't just for conscious awareness. Other second-order neurons also transmit this crucial data to your cerebellum.
Ah, for motor control.
Exactly. Absolutely vital for its role in motor coordination. Smooth movement depends on it.
That's a really key distinction right there. Unlike, say, vision or hearing, where the receptors are all packed into one organ.
Yeah, the eye or the ear.
Your somatosensory receptors are uniquely distributed throughout your entire body. You can feel things everywhere.
Which really highlights its fundamental importance, doesn't it? It's not localized.
Totally.
Yeah.
So how do we categorize this widespread system?
Well, that distribution helps us think about it in three broad divisions based on where the receptors are. First, and what we'll focus on a lot today, is the exteroceptive division.
Exteroceptive,
Yeah.
meaning outside.
Exactly. It's all about gathering info from the outside world, specifically through the contact of your skin with external objects. It includes receptors for touch, For pain, nociception is the technical term, and for temperature.
Okay, that makes sense. What are the others?
Then there's the proprioceptive component. Think of this as your body's internal GPS.
Ah, knowing where your limbs are without looking.
Precisely. It tells you about the position and movement of your limbs and body parts. Relies on receptors, mainly in your joints, muscles, and tendons. Absolutely essential for motor control, which is, like you said, a whole other fascinating deep dive itself.
Right.
And finally, the interoceptive division. This monitors the internal state of your body.
Like feeling full after a meal or needing the restroom.
Exactly. Mechanoreceptors detecting things like gut distension or bladder fullness. So these divisions give us windows into both our external world and our internal state.
And within those, we can group the actual sensations too, right?
Yes, into two major functional categories. First, you have fine or discriminatory touch sensations.
Okay, what falls under that?
Things like light touch, pressure, vibration, that feeling of flutter, which is like a low-frequency vibration in stretch or tension. Really precise stuff.
And the second category?
Pain and temperature sensations. This covers both noxious, meaning potentially harmful and innocuous cold and warm, as well as mechanical and chemical pain. And interestingly, itch is very closely related to pain. It actually travels along similar associated fiber pathways.
Fascinating. Okay, let's talk about the wires carrying these messages, the afferent fiber.
Right, the axons conveying these different somatosensory submodalities, they differ significantly in size and whether they have that myelin sheath, the fatty insulation that speeds up signals.
So faster signals for some sensations than others?
Definitely. For example, those quick, precise tactile sensations, they're primarily carried by large diameter myelinated fibers. The A class, very fast.
Okay, but pain and temperature?
That travels via much smaller diameter fibers, the lightly myelinated A fibers, and the completely unmyelinated C fibers. Much slower conduction.
And this difference, it's useful.
Incredibly useful, both for research and clinically, because you can selectively block or stimulate specific axon classes based on these properties.
Ah, so you can study different sensations in isolation.
Exactly. It helps us understand their unique roles and the underlying mechanisms. It's like isolating an instrument in an orchestra, like you said earlier, to hear its specific part. Really powerful for figuring things out.
Makes sense. Now, the skin itself, it's packed with receptors, right?
Oh, incredibly densely packed with specialized low-threshold mechanoreceptors. They respond to even very light touch.
So how do scientists study them?
Well, often they use a very fine rod or wire to gently press on tiny, localized regions of the skin and record the nerve activity. And they've observed two basic response patterns from the nerve fibers connected to these receptors.
Which are?
Fast adapting, or FA fibers, and slow adapting, or SA fibers. And they're present in roughly equal numbers, which is important for the richness of our touch perception.
Okay, fast adapting, they react quickly and then stop.
That's the gist. FA fibers give you a short burst of electrical activity right when a stimulus begins, and then they shut up, even if the pressure continues. They might give another little burst when the stimulus is removed.
And slow adapting, they keep going.
Right. SA fibers fire continuously, or at least increase their firing rate from the moment a stimulus starts until it ends. They signal the duration and steady intensity.
And these can be subdivided further based on receptive fields. What's that exactly?
Think of a receptive field as the specific patch of skin that, when stimulated, will trigger a response in that particular nerve fiber. It's like its listening area.
Okay.
And we have type 1 units and type 2 units. Type 1 units have very small receptive fields with really sharp, well-defined borders. Think about the pinpoint accuracy you have on your fingertips. They're highly sensitive to movement or edges cutting across their little field.
And type 2?
Much wider receptive fields. The borders are fuzzier, less defined. They usually have 1 point of maximum sensitivity, and it gradually fades as you move away.
So type 1 might cover just a tiny bit of a fingertip ridge.
Exactly. Well, a type 2 unit could cover most of an entire finger. This difference is absolutely critical for spatial resolution, telling things apart by touch.
Okay, so digging deeper, there are 4 main classes: FA1, FA2, SA1, and SA2.
That's right. And peripherally, these nerve axons can end in a couple of ways. Some terminate as simple free nerve endings, often wrapped around hair follicles. Others end within highly specialized encapsulated receptor structures, which are made up of supporting cells. These capsules modify the stimulus.
Right. So in hairless skin, like palms and soles, what do we find there?
Okay, in glabrous skin, SA1 afferents connect to Merkel cells right there in the basal epidermis, and FA1 afferents end in Meissner corpuscles, just below the epidermis.
And these have small fields.
Yes, both have small, precise receptive fields, great for fine detail. Then, deeper in the dermis and connective tissue, you find SA2 afferents ending in Ruffini endings and FA2 afferents ending in Pacinian corpuscles.
And these are the type 2 larger fields.
Exactly. Sensitive over a much larger territory.
You mentioned the capsules modify the stimulus. How does that work for the Pacinian corpuscles?
The Pacinian and Meissner ones are really clever. Their layered capsules act like mechanical filters. They actually dampen out slow, steady stimuli.
So they only respond to changes.
Precisely. It makes those afferents selectively sensitive only to changing stimuli, like vibration or the start and end of a touch. That's why they're fast adapting. It's like they're tuned to novelty.
That's neat. What about hairy skin? Is it the same setup?
Mostly similar for SA1 and SA2, Merkel and Ruffini endings are there. Pacinian corpuscles contribute to FA2 afferents too, but they're deeper down, not right in the hairy skin itself.
So no direct equivalent of the Meissner corpuscle?
Not exactly. Instead, hairy skin has these hair units. These are afferents whose free endings wrap around hair follicles. Each one connects with maybe 20 hairs or so.
Creating a large receptive field.
Yes, large and very sensitive. They're exquisitely sensitive to even the slightest movement of a single hair. You also have field units, which broadly respond to skin touch with large fields.
This gets us to perception and spatial acuity, right? How sensitive are these things, really?
It's quite remarkable. Think about this. A single electrical spike, just one action potential from an FA1 efferent in your finger. You can actually consciously perceive that.
Wow. Just one spike.
Just one. It tells us that, at least for supersensitive areas like fingertips, the sensitivity you experience is primarily determined right there by the peripheral receptors.
But not everywhere.
Not quite. For other skin regions, your perception is more influenced by central nervous system factors, like how much attention you're paying.
This precision explains things like the 2-point discrimination test, doesn't it?
Absolutely. Where a doctor touches you with 2 points, you feel them as distinct only if they're separated by a certain minimum distance.
And fingertips are best at that.
By far. Shortest threshold distance. Why? Because it primarily depends on that high density and the small receptive fields of the type 1 units we discussed, the Merkel and Meissner related ones.
So density equals sensitivity?
Largely, yes. The density of type 1 units diminishes as you go from fingertip to palm to forearm, and that correlates perfectly with the increasing distance you need to tell 2 points apart. It's a beautiful map of innervation density to function.
What about coding the quality of a stimulus? Like, how do we know if it's pressure versus vibration?
Good question. If researchers stimulate a single SA fiber electrically, you'd feel a sensation of steady pressure right in that fiber's receptive field. If they crank up the frequency of the pulses, the perceived pressure increases.
So firing rate codes for force in SC fibers.
Exactly. Now, if you repetitively stimulate an FA fiber, you'd first feel a tapping sensation. Then, as the frequency increases, it turns into a feeling of vibration.
Okay. Different qualities coded by different fiber types and firing patterns.
Right. And here's a really critical insight. In neither case does stimulating just that one fiber class suddenly make you feel, say, pain.
So pressure stays pressure, vibration stays vibration.
Exactly. As long as only that specific fiber class is activated, this provides really strong evidence that pain isn't just an intense version of touch. It's a distinct submodality. It has its own dedicated fibers and specific nerve cell populations in the CNS.
Got it. Moving beyond the skin receptors now to the broader body, how do these signals get into the central nervous system?
Right. The axons from your peripheral nervous system enter or leave the CNS via spinal roots, or through cranial nerves for the head and neck. Each dorsal root of a spinal segment is entirely made up of the central processes of its associated dorsal root ganglion cells.
And the ventral root is motor.
Primarily motor axons, yes, leaving the spinal cord.
Let's talk about dermatomes. That's the nerve map on the skin, right?
Exactly. It's laid out during embryonic development. Each dermatome is a specific area of skin supplied by a single dorsal root ganglion.
They look a bit weird in humans, though.
They do appear distorted because of how our limbs develop and our upright posture. But the pattern becomes remarkably clear if you imagine a person in a quadrupedal stance, like on all fours.
Is there a clinical example that shows this clearly?
Oh, a classic and often painful one is shingles, herpes zoster.
Yeah.
It's the reactivation of the chickenpox virus, which lies dormant in a dorsal root or cranial nerve ganglion. When it reactivates, it infects the cells of that one ganglion.
And the rash follows the dermatome.
Perfectly. You get a painful or itchy rash, strictly confined to that dermatome on one side of the body, ending sharply at the midline, often looks like a belt or stripe. It's a living map of that nerve's territory.
Wow. And within the dorsal roots themselves, are the fibers organized?
They are. The large myelinated primary afferent fibers, the fast-tex ones, are positioned more medially.
Towards the middle?
Right, while the smaller myelinated and unmyelinated fibers, pain, temperature, are more lateral, towards the side. The large medial fibers then enter the dorsal column of the spinal cord.
And they split?
Yes, into branches that ascend upwards towards the brain, and branches that connect locally within the spinal cord gray matter for reflexes and local processing.
Okay. What about the face? Is it a similar setup?
Quite similar, yes, mainly managed by the trigeminal nerve. Nerve fibers from the trigeminal ganglion pass through its three main divisions: ophthalmic, maxillary, mandibular, innervating dermatome-like regions of the face.
And it carries touch and pain temperature?
Both, plus sensation from your teeth, oral and nasal cavities, and even the dura mater, the covering around your brain.
Where do these trigeminal fibers go in the brainstem?
Their central processes enter the brainstem at the mid-pons level. This is where the main sensory trigeminal nucleus is. Some axons, especially the larger ones for fine touch and vibration, synapse right there.
And the others? Pain and temperature.
Those, particularly the smaller fibers, descend further down to synapse in the spinal trigeminal nucleus.
Now, you mentioned proprioception body position sense. How does that work for the head? Is it unique?
It is. Here's a really unique detail. The cell bodies of the first-order proprioceptive fibers for the head are actually located within the CNS itself.
Not in a ganglion outside.
Nope. They're in the mesencephalic portion of the trigeminal nucleus. These neurons specifically innervate the stretch receptors in your mastication muscles, your jaw muscles.
Huh. And where do they project?
They project to the trigeminal motor nucleus for local reflexes, like the jaw jerk reflex, and also to the reticular formation and the chief sensory trigeminal nucleus. Very unusual arrangement.
Okay, now let's trace these signals as they travel the information superhighways to the brain. You said they stay segregated.
That's a key point for understanding this at a deeper level. Different somatosensory sensations continue to travel in separate pathways within the CNS. They ascend the spinal cord and brainstem via distinct routes.
So fine touch goes one way.
Right. Fine discriminatory touch is conveyed by the dorsal column medial lemniscus pathway.
And pain, temperature, crude touch.
That's carried by the anterolateral system. We'll get into that one soon.
And proprioception.
Proprioceptive information travels via routes that partly overlap with the dorsal column path, but also via specific spinocerebellar pathways heading to the cerebellum. This functional segregation is largely maintained, although there's some redundancy. Losing one pathway doesn't always mean complete loss of that sensation type.
Okay, let's focus on that dorsal column medial lemniscus pathway. Critical for discriminatory touch and proprioception. Where does it start?
It begins with the ascending branches of those large myelinated axons from the first-order dorsal root ganglion cells. These form the dorsal columns in the spinal cord.
Traveling up.
All the way up to the caudal medulla, the lower part of the brainstem.
And there they synapse.
Yes, in specific dorsal column nuclei. The nucleus gracilis receives information from the lower body, and leg gets more medial. And the nucleus communis receives information from the upper body, and arm it's more lateral.
And there's a map here, too.
Absolutely. A precise somatotopic map that's maintained all the way up. Legs are most medial, then trunk, then upper limb is added laterally as you go up the spinal cord. It's a consistent body map.
So the second-order neurons are in these medullary nuclei. How do they compare to the first-order ones?
Their responses are similar, but with a couple of key differences. Their receptive fields are usually a bit larger.
Why is that?
Because multiple primary afferents converge onto each second-order neuron. And for the same reason, they sometimes respond to more than one type of sensory receptor. A bit of integration starts here.
Okay. Now, here's a critical point. You mentioned the crossing over, the decussation.
Yes, this is vital. The axons of these second-order neurons in the dorsal column nuclei cross the midline right there at the same medullary level where their cell bodies are.
And after crossing,
They form a bundle called the medial lemniscus, which then projects upwards straight to the thalamus.
why is this crossing point so important clinically?
Because if you have damage to this pathway below the crossing anywhere in the spinal cord, you lose fine touch and proprioception on the same side of the body as the lesion. It's a lateral deficit.
Okay.
But if the damage is above the crossing, in the brainstem or higher, the deficit will be on the opposite side of the body, contralateral. And because the map is so precise, A localized lesion can cause selective loss just in, say, an arm or a leg. It
Right.
helps pinpoint the problem.
Makes sense. And the third order neurons in this pathway, where are they?
They're located in the ventral posterior lateral VPL nucleus of the thalamus.
And from the VPL.
They project up to the somatosensory areas of the cerebral cortex, mainly SI. This whole pathway is absolutely essential for your fine touch discrimination, vibration sense, recognizing object shapes by feel, reading braille, all that precise stuff.
How would you test for problems here?
Clinically, we look at things like graphesthesia. Can you recognize a letter traced on your skin? Or can you tell the direction of a line drawn across your skin? Stereognosis, recognizing objects by touch alone. These rely heavily on this pathway.
Is it an all-or-nothing pathway, though?
Not entirely. It's important to note that some basic tactile awareness and localization can remain even if the dorsal columns are completely lost. Other ascending pathways provide some backup.
But pain and temperature are fine if this pathway is damaged.
Correct. Cutaneous pain and temperature are unaffected because they travel in the anterolateral system. Interestingly, though, visceral pain from internal organs is often diminished by dorsal column damage.
Okay. And for fine touch from the face, is it a similar route?
Very analogous, yes. Primary afferents from the face, teeth, oral nasal cavities, cranial meninges, synapse in the main sensory nucleus of the trigeminal nerve in the pons.
All right.
This nucleus then relays that tactile information to the contralateral ventral posterior medial VPM thalamic nucleus via the trigeminothalamic tract.
VPM for face, VPL for body. Got it.
Exactly. And then the third order neurons in the VPM project straight to the face area of the somatosensory cortex. Same logic, different nucleus.
So let's bring proprioception back in. Body awareness. Where does that info go?
Right. The proprioceptors are constantly updating your brain about body part positions and movements. This info goes mainly to two places.
Cerebellum and cortex.
You got it. Cerebellum uses it for coordinating smooth movements. Cerebral cortex uses it for your conscious awareness of where your body parts are. That's kinesthesia.
Are there specific pathways for the cerebellum?
Yes. Major ones carrying both cutaneous and proprioceptive info. For the trunk and lower limb, dorsal root ganglion cells synapse in Clark's nucleus in the spinal cord, levels T1L2. Their axons ascend in the dorsal spinocerebellar tract and enter the cerebellum via the inferior cerebellar peduncle.
And for the upper limb?
Dorsal root ganglion fibers from the cervical levels ascend in the cuneate fasciculus up to the external cuneate nucleus in the medulla. Axons from there form the cuneocerebellar tract, also entering the cerebellum via the inferior cerebellar peduncle. Keeps the cerebellum fully informed.
What about head proprioception to the cerebellum?
That input is carried by those unique cells in the mesencephalic nucleus of the trigeminal nerve. Remember the first-order neurons for jaw muscle stretch receptors?
Right, the ones inside the CNS.
Yes. Their central processes project partly to the trigeminal motor nucleus for reflexes, but also to the reticular formation. Axons from there join the trigeminothalamic tract to the VPM thalamus. And they're also direct trigeminocerebellar pathways carrying various somatosensory info from the head to the cerebellum.
Okay, moving up to the brain's master processors. Let's talk thalamus, the sensory gateway.
Right. The main destination for ascending somatosensory info is the ventroposterior nuclear complex of the thalamus. This includes the VPL, the VPM, and a smaller VPI nucleus.
And the inputs are the medial lemniscus to VPL.
For the body, yes, and the trigeminothalamic tract from the main sensory nucleus of the trigeminal to VPM for the face. Plus, these nuclei also receive the bulk of the pain and temperature information coming up the spinothalamic tract, or its trigeminal equivalent.
How do the neurons here behave?
It's insightful, actually. Their responses often mirror the first and second order neurons further down, but their receptive fields are slightly larger, and they're usually dominated by input from just one type receptor, FA, SA, or proprioceptive. Still quite specific.
Is the thalamus just passing signals along?
Oh no, it's actively regulated. There are networks of inhibitory neurons, GABAergic interneurons. And importantly, the reticular nucleus of the thalamus, this thin shell surrounding the main thalamus, is mostly inhibitory.
Like gatekeepers.
Exactly. They project back to the VPL, VPM, and other nuclei, helping to fine-tune and filter the sensory information that finally gets passed up to the cortex.
And this relates to sleep and wakefulness.
Absolutely. The excitability of these thalamic neurons changes dramatically with your sleep-wake cycle. During drowsiness or under anesthesia, they can fall into this bursting pattern of firing.
Bursts.
Yeah, rhythmic bursts. These intermittently excite neurons in the cortex, leading to those characteristic alpha rhythms or spindling patterns you see on an EEG. It reflects how inhibition from places like the reticular nucleus is gating the flow of information. It's a key part of modulating consciousness.
And the body map in the thalamus. Still there.
Definitely. Thalamic neurons always have receptive fields on the opposite contralateral side of the body. And the map is systematic. Lower limb is most lateral, upper limb medial in VPL. Head even more medial in VPM.
But you said multiple maps.
Yes, it seems there are separate overlapping somatotopic maps for different submodalities, touch, proprioception, pain within this complex.
How are they arranged?
Well, evidence suggests cutaneous input, skin, drives cells in a central core region of VPL-VPM, while proprioceptive information, muscles, joints, goes to cells forming a shell around this core. It's spatially organized by modality, too.
Does all sensory info go just to VPL, VPM?
Not quite. The spinothalamic tract, carrying pain and temperature, also projects to other thalamic regions, like the posterior nucleus and the central lateral nucleus of the intralaminar complex.
Intralaminar. What's their role?
These aren't neatly mapped like VPL, VPM. They project diffusely to broad areas of the cortex and the basal ganglia. They seem to be involved more in arousal, attention, maybe the emotional aspect of pain.
Okay, finally reaching the top level, the somatosensory cortex, the conscious interpreter.
Right, those third-order sensory neurons in the VPL and VPM project directly up here. The main receiving areas are SI, the primary somatosensory cortex, right on the post-central gyrus.
Just behind the central sulcus.
Exactly. And STI, the secondary somatosensory cortex, tucked into the superior bank of the lateral fissure.
And the body map persists here too, the homunculus.
Yes, both SI and SCOI have detailed body maps. The most famous is that sensory homunculus on the post-central gyrus in SI.
The distorted little man picture.
That's the one. It's distorted because the amount of cortical real estate dedicated to a body region is proportional to its innervation density and sensory importance.
So hands, lips, tongue are huge.
Exactly. They take up disproportionately large areas compared to, say, your back or legs, reflecting how crucial they are for exploring the world through touch.
Does SI have subdivisions?
It does. Functionally and morphologically distinct subdivisions called Brodmann areas,
Yeah.
3A, 3B, 1, and 2, each has its own complete somatotopic map.
Do they process different things?
Seems so. Cutaneous input from the skin dominates in areas 3B and 1, while muscle and joint input proprioception dominates in areas 3A and 2, so even within SI you have specialization for tactile versus proprioceptive processing.
You mentioned columnar organization earlier. Does that apply here?
Yes, SI cortex has a strong columnar organization. Neurons stacked vertically, perpendicular to the surface, tend to have very similar response properties and receptive fields.
Like processing units.
Sort of. And nearby columns can process info for different modalities. For instance, one column in area 3B might get mostly FA input, while its neighbor gets SA input from the same skin area. It allows for parallel processing of different features.
Is SI just receiving signals or doing more?
Definitely doing more. It's not just a passive receiver, it's actively involved in higher order processing, starting to extract specific features from the sensation.
Like what?
Well, for example, some neurons in area 1 respond best to a stimulus moving in one specific direction across the receptive field, but not the opposite direction.
Ah, detecting movement direction.
Exactly. These specialized neurons likely contribute to recognizing stimulus direction, and they're probably vital for things like detecting if an object you're holding is starting to slip. Feature extraction begins here.
So what happens if there's damage, a lesion in SI?
It typically produces sensory changes similar to a thalamic lesion, but usually confined to the part of the homunculus map where the damage is.
So it depends where the lesion is.
Right. Damage to the lateral cortex might affect the face. Damage medially might affect the leg. The biggest deficits are usually in discriminative touch and position sense.
So graphesthesia and stereognosis would be impaired.
Severely impaired, yes. Recognizing objects by touch becomes very difficult. Pain and temperature might be relatively okay, but large cortical lesions can sometimes even cause a central pain state similar to thalamic pain.
Okay, let's shift gears now to pain and temperature. You said they travel together but are distinct from touch.
Absolutely critical point. They are mediated by overlapping sets of receptors and nerve fibers. But pain is not simply what happens when you stimulate touch pathways more strongly. If you experimentally zap SA afferents harder, you just feel more pressure, never pain. It's qualitatively different.
And the fibers involved are slower.
Primarily, yes. The relatively slow-conducting A, lightly myelinated, and C, unmyelinated fiber classes carry most pain and thermal sensations.
But not all A and C fibers.
Correct. It's important to remember some atypical fibers in these classes actually respond to light touch, behaving like low-threshold mechanoreceptors. It's not perfectly clean segregation by fiber class alone.
And their endings are different from touch receptors.
Morphologically, they mostly originate as free nerve endings. They don't have those elaborate capsules like Meissner or Pacinian corpuscles, although they are still partially covered by Schwann cells.
But they're still specialized.
Functionally, yes. Despite looking simpler, these A and C axons form a diverse population. Each is differentially sensitive to various tissue-damaging stimuli, mechanical, thermal, or chemical. These specialized receptors are called nociceptors.
Noci meaning harm.
Exactly. They're dedicated to detecting stimuli that are potentially harmful or damaging to tissues.
And they can become sensitized.
That's a key distinction. Nociceptors can become sensitized, meaning their threshold lowers and they respond more vigorously. And fascinatingly, a significant number of C fibers are normally silent. They don't respond to any stimuli until they are first sensitized, perhaps by inflammation or injury. They get woken up by damage.
This leads to the idea of first and second pain, right, based on the fiber speeds.
Precisely. The pain system uses the faster A and slower C axons to create two distinct sensations. AL fibers conduct faster and are thought to underlie first pain.
That initial sharp pricking feeling.
Yes, sharp pricking, usually well localized, happens almost instantly. Then the C fibers, being slower, are responsible for second pain.
The duller burning ache that comes later.
Exactly. Duller, more diffuse, often described as burning or aching, and it lingers. Experiments selectively activating these fibers confirm it. A stimulation feels like first pain. C fiber stimulation feels like second pain.
And within these A and C fibers, there's more diversity.
Oh, yes. They vary hugely in sensitivity. Some respond to low-threshold mechanical stimuli, others only high-threshold. Some are insensitive until sensitized. Thermally, you have specific fibers responding best to warmth. Noxious heat, above about 43 degrees C, cool or noxious cold, below about 15 degrees C.
And chemical triggers too.
Yep, tested with irritants like capsaicin from chili peppers, mustard oil, acids.
So fibers can be sensitive to multiple things.
Many are. We classify them based on what they respond to. For example, C fibers, sensitive only to strong mechanical stimuli, are C mechanosensitive. Those sensitive to both heat and mechanical stimuli are C mechanoheat sensitive, often called polymodal fibers. They're very common, maybe half of all cutaneous C fibers.
What other types are important?
You have A and C cold-sensitive fibers, A and mechanosensitive or mechanoheat-sensitive fibers. And interestingly, the second most common type of C fiber is actually the mechanoheat-insensitive afferent.
The silent ones.
Likely. They don't respond to noxious stimuli until they get sensitized by injury or inflammation. This really highlights how dynamic the pain system is. It changes based on context.
Okay, so these signals reach the spinal cord. Where do they connect?
The central axons of A and C fibers carrying pain and temperature from the body terminate in the dorsal horn of the spinal cord gray matter. But they target specific layers, or laminae.
Different targets for A and C?
Yes. A fibers primarily target laminae I, V, and maybe X. C fibers mainly terminate in the very superficial laminae I and II, also called the substantia gelatinosa. These distinct landing zones strongly suggest different processing pathways right from the first synapse.
This relates to the gate control theory, doesn't it? How rubbing a sore spot helps.
Exactly. It's a crucial concept. Innocuous stimuli, like rubbing or vibration, can reduce or even block pain signals.
How does that work at the cellular level?
The theory proposes that the large diameter A and A fibers, the fast touch fibers, activate inhibitory interneurons within the dorsal horn. These interneurons then release inhibitory neurotransmitters, like GABA, and GABA dampens the activity of the spinothalamic tract cells, the ones that send the pain signal up to the brain. It does this both presynaptically, reducing transmitter release from the pain fibers, and postsynaptically, directly inhibiting the projection neuron.
So touch signals can effectively close the gate on pain signals heading up.
That's the core idea, a built-in mechanism for modulating pain right at the entry point.
What about head pain? Toothaches, headaches?
Similar process, but involving the trigeminal system. Nociceptive and thermoreceptive info from the head enters the brainstem mainly via the trigeminal nerve, though some comes via facial, glossopharyngeal, vagus nerves too. These fibers then descend through the brainstem in the descending tract of the trigeminal nerve, and they synapse on second-order neurons in the descending nucleus of the trigeminal nerve. This nucleus is essentially the brainstem equivalent of the spinal cord's dorsal horn for head pain.
Is there a clinical condition related to this trigeminal pain pathway?
Oh, a particularly nasty one is trigeminal neuralgia, tic douloureux.
What's that like?
It's characterized by sudden attacks of severe, stabbing electric shock-like pain in the distribution of one or more branches of the trigeminal nerve, often triggered by very light touch, like wind on the face, chewing, or talking.
What causes it?
It's often linked to mechanical irritation of the trigeminal ganglion or nerve root, frequently by a pulsating artery pressing against it. It seems to cause abnormal firing in the nerve. Remarkably, surgically moving the offending artery away from the nerve can often resolve the pain completely.
Wow. Okay, so the pain signal has reached the second-order neuron in the spinal cord or trigeminal nucleus. Where does it go next? What are the central pain pathways?
There are several important ascending pathways. The most crucial one for conscious perception of somatic pain and temperature from the body is the spinothalamic tract. It also carries some basic tactile info.
Where do its neurons originate and where do they cross?
The second-order neurons originate mainly in the spinal cord dorsal horn laminae auro, and deeper ones like 4V6. And critically, their axons cross to the opposite side of the spinal cord, usually within a few segments of their origin.
So they cross low down, unlike the dorsal column pathway.
Exactly. A very important distinction. These crossed axons then ascend in the ventrolateral funiculus, or anterolateral quadrant, of the spinal cord,
All the way up.
through the brainstem, all the way to the thalamus, where they predominantly terminate in the VPL nucleus, just like the fine touch pathway, but carrying different information.
And from the thalamus to the cortex.
Yes. Nociceptive signals are forwarded from VPL, not just to the somatosensory cortex, SI, for sensory discrimination, telling you where the pain is, what it feels like, but also to other cortical areas heavily involved in effective and motivational responses. Think places like the cingulate gyrus and the insula, which are part of the limbic system.
Which explains the emotional component of pain. The unpleasantness, the fear.
Precisely. Pain isn't just a sensation. It's an experience with a powerful emotional dimension, driven by these projections to limbic-related areas.
What kinds of cells are in the spinothalamic tract? Do they only respond to pain?
Good question. Many do receive excitatory input from nociceptors. A major type is the wide dynamic range, WDR cell, or multireceptive cell.
Wide dynamic range? Meaning?
They respond weakly to light tactile stimuli, but they respond much more strongly, often with accelerating firing rates, to noxious stimuli. So their main job is signaling noxious events, but they can detect touch.
Could that cause problems?
Yes. Under certain pathological conditions like nerve injury or inflammation, these WDR neurons can become hypersensitive or centrally sensitized. In that state, even normal tactile stimuli can trigger a strong response in them, leading to the perception of pain from light touch. This is thought to be a key mechanism behind mechanical allodynia.
Where touch hurts.
Exactly. Other spinothalamic cells are more specific, high-threshold or nociceptive-specific cells. They are activated only by noxious stimuli. They don't respond to normal touch at all.
This convergence. Does it relate to referred pain, like heart attack pain felt in the arm?
Absolutely. Referred pain is a fascinating phenomenon where visceral pain from internal organs is perceived as coming from a cutaneous skin site.
Why does the brain get confused?
It happens because the spinothalamic tract neurons that receive input from visceral nociceptors also typically receive converging input from cutaneous receptors in a specific dermatome.
So the brain can't tell if the signal originated in the organ or the skin.
Essentially, yes. Since cutaneous stimulation is far more common, the brain tends to interpret the signal as coming from the more frequently stimulated skin area. So heart muscle ischemia gets referred to the chest wall and left arm dermatomes, which share spinal cord segments with the heart's sensory input.
What neurotransmitters are involved in these pain synapses?
It's complex.
Yeah.
At the first synapse in the dorsal horn, nociceptive afferents release glutamate as the primary fast excitatory transmitter, acting on AMPA receptors.
Okay.
With strong or repetitive stimulation, glutamate also activates NMDA receptors, which can lead to longer-lasting changes and increased excitability, part of that central sensitization process.
Are there other transmitters?
Yes, peptides play a big role as neuromodulators, things like substance P, SP, CGRP, VIP. Substance P, released along with glutamate, seems particularly important for inducing central sensitization, making those spinothalamic cells hypersensitive for potentially long periods.
What about turning the pain signal down? Inhibition.
Crucial. Spinothalamic tract cells can be inhibited by local interneurons releasing GABA and glycine, and also by descending pathways releasing monoamines like serotonin and norepinephrine, and, very importantly, endogenous opioid peptides like enkephalins.
Your body's own painkillers.
Exactly. What's intriguing is that the inhibitory receptive fields controlling these cells can be huge, sometimes covering most of the body.
How could that be useful?
Well, it might help explain how diffuse stimuli or manipulations far from the pain site, like TNS, transcutaneous electrical nerve stimulation, or maybe even acupuncture, could potentially suppress pain by activating these widespread inhibitory systems.
Are there other ascending pain pathways besides spinothalamic?
Yes, definitely. There's the spinal-reticular tract. Dorsal horn neurons project up to the reticular formation in the brainstem, medulla, and pons.
What's different about these neurons?
They often have very large, sometimes bilateral receptive fields and respond well to noxious stimuli. The reticular formation neurons they activate, especially norepinephrine-producing ones, then project up to the intralaminar nuclei of the thalamus.
The ones involved in arousal and attention.
Exactly, and then diffusely to the cortex. So this pathway is heavily involved in the arousal and attentional aspects of pain, making you pay attention when something hurts. It also feeds into descending pain control systems.
Okay, what else?
There's also the spinomesencephalic tract. Many of these cells also respond to noxious stimuli. They terminate in midbrain nuclei, including the periaqueductal gray.
That sounds important.
It's a key command center for your body's own pain control system, the endogenous analgesia system. Activation of this pathway can trigger strong motivational responses, think vocalizing, trying to escape, and the info is relayed from the midbrain to the thalamus and the amygdala.
Amygdala for emotion.
Yes. This forms a critical pathway for noxious stimuli to trigger emotional responses, like fear and aversion associated with pain.
And for facehead pain, are there similar pathways going up from the trigeminal nucleus?
Yes, analogous ascending pathways exist. Neurons in the trigeminal nucleus transmit pain and temperature info primarily to the contralateral VPM thalamus via the descending trigeminothalamic tract, which runs close to the medial lemniscus. They also project to those intralaminar nuclei for the arousal attention component. And from the thalamic nuclei, VPM and intralaminar, projections go to the somatosensory cortex for discrimination and to other cortical regions like cingulate, insula, for the motivational affective responses. Same logic, different anatomical names.
What happens clinically if you interrupt these pathways? The spinothalamic tract.
Historically, a procedure called anterolateral cordotomy was performed. It surgically cuts the spinothalamic tract and nearby pathways in the ventrolateral spinal cord.
What was the effect?
It leads to a loss of pain and temperature sensation on the contralateral side of the body below the level of the cut. Both the sensory discriminative part, knowing where it hurts, and the motivational affective part, the unpleasantness, are reduced.
Was it effective?
It was used for severe intractable pain, like from cancer, but it's much less common now. Better drug therapies exist, and often the pain would unfortunately return months or years later. Also, it causes loss of cold-warm sensation, but tactile sensation is usually pretty intact because the dorsal columns are spared.
Can damage higher up, like in the thalamus, cause pain issues?
Yes, and paradoxically, it can cause more pain, not less. Lesions in the somatosensory thalamus, VPL, VPM, often from a stroke, can result in a devastating condition called central pain, sometimes specifically thalamic pain.
What's that like?
It's characterized by severe spontaneous burning or aching pain in the affected body parts. And often, patients experience extreme allodynia. Even the slightest touch becomes intensely painful, way below the normal threshold for pain receptors.
Why would damage cause pain?
The mechanisms aren't fully understood, but one leading hypothesis involves abnormal plasticity or rewiring after the lesion. Maybe surviving low-threshold touch fibers from the dorsal column system sprout and make new inappropriate connections onto thalamic neurons that normally only signal pain. Similar central pain states can also result from lesions in the brainstem or cortex affecting these pathways.
This seems related to neuropathic pain. What defines that?
Neuropathic pain is fundamentally different because it's pain that occurs in the absence of ongoing nociceptor stimulation. It's not driven by tissue damage activating pain receptors. It's caused by damage or dysfunction within the nervous system itself, either peripheral nerves or CNS structures involved in processing pain.
Can you give some examples?
Sure. In the periphery, one type is causalgia, also known as complex regional pain syndrome type 2. It's a severe burning pain, often with allodynia, that can develop after a traumatic injury to a peripheral nerve.
What causes the pain if the injury is healing?
It's thought to involve spontaneous abnormal activity firing off in the damaged dorsal root ganglion cells, maybe due to changes in ion channels, like sodium channels. Interestingly, in some cases, this pain seems to be maintained by the sympathetic nervous system.
How so?
because blocking sympathetic nerve activity to the limb can sometimes relieve the pain. The idea is maybe damaged sympathetic fibers sprout aberrantly into the DRG, or adrenoreceptors get upregulated on sensory neurons, making them sensitive to norepinephrine released by sympathetic nerves.
Another really striking example is phantom limb pain, right?
Absolutely. This occurs after amputation. The person feels vivid, often excruciating pain in the missing limb. It's clearly not caused by active nociceptors in the limb because the limb isn't there anymore.
So the brain is generating the pain itself.
It seems to be a result of complex maladaptive changes in the central representation of that limb within the spinal cord and brain. A profound example of how central processing can create sensation without peripheral input.
Is there central neuropathic pain, too?
Yes, this occurs after damage to CNS structures themselves, like the thalamus or the spinothalamic cortical pathway, as we discussed with thalamic pain. It's often a severe, spontaneous pain. But paradoxically, the same lesion might also reduce pain evoked by peripheral stimulation. It's complex, likely involving widespread changes in neuronal activity and connectivity far beyond the initial injury site.
Okay, let's dive really deep now into the molecular details. Transduction in the somatosensory system. How do these stimuli actually get converted into electrical signals at the nerve endings?
Right, this is where the magic happens at the protein level. The general principle is that at the specialized endings of these peripheral axons, specific ion channels are activated by the relevant stimulus mechanical force, heat, cold, or chemicals.
And channel opening lets ions flow.
Exactly. This flow of ions, usually positive ions like sodium or calcium flowing in, creates a change in the electrical potential across the membrane. That's the generator potential. If it's large enough, it triggers voltage-gated sodium channels nearby to fire action potentials down the axon.
Is it just that simple channel opening?
Well, it's a bit more complex. Those generator potentials are definitely shaped and modified by other voltage-gated channels present in the nerve-ending excitatory sodium and calcium channels amplifying it, inhibitory potassium channels dampening it. And in those encapsulated terminals, like Pacinian corpuscles or Merkel cell complexes, the accessory structures themselves play a big role. The capsule of the Pacinian corpuscle mechanically filters the stimulus, remember?
Right, filtering out steady pressure.
And in the Merkel cell case, it's even more interactive. Both the Merkel cell and its associated nerve ending have mechanosensitive channels. Plus, the Merkel cell itself releases neurotransmitters onto the nerve ending when it's compressed, actively contributing to the signal. It's a partnership.
Identifying these specific channel proteins must be crucial for understanding touch and pain, and maybe finding treatments.
Absolutely vital. For mechanotransduction, for a long time, people thought ASIC proteins, acid-sensing ion channels, might be the key, partly based on work in invertebrates.
But that turned out not to be the case.
Largely, no. Experimentally knocking down ASIC proteins in mammalian dorsal root ganglion cells doesn't really affect the main mechanoactivated currents. So ASICs might play some modulatory role, but they're not the primary channel for cutaneous touch, at least not the rapid ones.
So what is the key protein for touch?
The big breakthrough has been the identification of piezo-2. This protein is now widely considered the channel underlying transduction for cutaneous rapidly adapting mechanical responses.
What makes scientists so sure about piezo-2?
Several lines of evidence. It forms a non-selective cation pore that directly opens in response to mechanical stimuli. Its activation and inactivation kinetics, how quickly it opens and closes, perfectly match those rapidly adapting currents recorded from neurons.
Okay.
It's blocked by specific pharmacological agents known to block these currents, like gadolinium and ruthenium red. Crucially, if you use techniques like Serenade to specifically block piezo-2 expression in DRG cells, the rapid mechanoactivated current disappears. And piezo-2 mRNA is clearly expressed in the right place, dorsal root ganglion cells. It ticks all the boxes.
Does piezo-2 only handle light touch?
No, and this is really important. It's expressed in both low-threshold mechanoreceptors for light touch and in nociceptive-type ganglion cells. This indicates it likely plays a role in sensing both innocuous and painful mechanical stimuli.
Wow. So relevant for both normal touch and mechanical pain.
Seems so.
Yeah.
And piezoproteins, piezo-1 is another family member, are found in various organs throughout the body, suggesting they might underlie visceral sensation too, like stretch receptors in the bladder or gut.
You mentioned proprioception earlier.
Yes. Critically, piezo-2 has also been identified as the main mechanotransducer in proprioceptors, the sensors in your muscle spindles and Golgi tendon organs that tell your brain about muscle stretch and tension. So it's truly a foundational protein for sensing our body's physical interaction with the world and its own state.
Okay, what about thermal transduction? How do we sense hot and cold at the molecular level?
A landmark discovery here was cloning the receptor for capsaicin.
The stuff that makes chili peppers hot.
Exactly. They found the protein that binds capsaicin and causes that burning heat sensation. And importantly, this receptor, or related proteins, are expressed in specific populations of dorsal root ganglion cells that respond to heat.
And this led to a whole family of channels.
We did. These proteins belong to the TRP family, which stands for transient receptor potential.
Weird name. Where did that come from?
From early studies in fruit flies, actually. A mutation in the founding member of this gene family caused photoreceptor cells to have only a transient response to continuous light, instead of a sustained one. The name stuck.
What do TRP channels do in mammals?
They're incredibly diverse. There are about 27 known TRP genes in mammals, grouped into 7 subfamilies. They're involved in all sorts of sensory processes, phototransduction in the eye, chemotransduction for taste and smell, even some mechanoreceptors. Structurally, they typically form cation-permeable channels, often resembling voltage-gated potassium channels.
But some are specifically temperature sensors.
Yes, this is the really exciting part. Specific TRP proteins act as molecular thermometers, each activated within a distinct temperature range. Together, they cover the entire physiological spectrum from cold to burning hot.
Can you give some examples?
Sure. TRPV1, the capsaicin receptor, is activated by temperatures above about 42 degrees C, that's noxious heat. TRPV2 kicks in at even higher, potentially damaging temperatures above 52 degrees C.
What about warmth?
TRPV3 responds in the warm range, around 34-38 degrees C, and it's also activated by camphor. TRPV4 senses temperatures between about 27-40 degrees C.
And for cold?
TRPM8 is the main cool-cold sensor, activated by temperatures below about 25 degrees C, and it's also activated by menthol, giving you that cooling sensation from mint. And TRPA1 responds to even colder temperatures, below about 18 degrees C, and is also activated by pungent compounds like mustard oil and wasabi, contributing to that noxious, burning cold feeling.
Do these channel ranges match the responses of actual temperature-sensing nerve fibers?
For the heat-sensitive fibers, the match is pretty good. The response ranges align well with the activation thresholds of individual TRP channels, like TRPV1, 2, 3, and 4.
And cold fibers?
Cold fibers often respond over a broader range than any single channel like TRPM8 or TRPA1. This suggests that individual cold-sensitive neurons might express multiple types of TRP channels, allowing them to integrate signals across a wider temperature spectrum. It's an elegant molecular system for detecting thermal changes.
Okay, finally, let's talk about modulation and centrifugal control. How is sensation actively shaped or filtered? Let's start peripherally.
Right. Modulation of the transduction process itself. A key concept here is peripheral sensitization, especially regarding pain. When nociceptors are activated by tissue damage, they don't just send signals up. They also release chemicals locally at the injury site.
Like what chemicals?
Things like tachykinin, substance P again, CGRP, bradykinin, prostaglandins. This release causes what's called neurogenic inflammation, local swelling, redness, increased blood flow.
But does it affect the nerves themselves?
Yes, crucially. This inflammatory soup can directly act back on the nociceptor endings, including those previously silent nociceptors we talked about. It lowers their activation threshold and it increases their responsiveness. They become sensitized.
Making the area more sensitive to pain.
Exactly. This peripheral sensitization is thought to be a major contributor to clinical phenomena like hyperalgesia, where something that was already painful now hurts even more. And also allodynia, where a normally non-painful stimulus like light touch on sunburned skin now causes pain.
Are specific molecules involved in sensitizing the channels?
Yes. For example, thermal hyperalgesia, increased sensitivity to heat, is strongly linked to the sensitization of the TRPV1 channel by inflammatory mediators. And substances known to cause mechanical hyperalgesia and allodynia have been shown to increase the current through piezo2 channels, implicating piezo2 sensitization in those conditions too.
So the periphery can definitely change its sensitivity. What about control coming down from the brain? Centrifugal control.
This is absolutely critical for understanding sensory perception. Your experience isn't just a passive readout of what's coming in. It often involves active exploration, moving your hand to feel texture, scanning with your eyes. Sensory information is frequently received because your motor system initiated an action.
Okay.
More than that, the transmission of signals within the sensory pathways themselves is powerfully regulated by descending control systems originating in your brain.
The brain filtering its own input.
Precisely. These systems allow the brain to modulate the flow of sensory information. effectively filtering incoming messages. It lets you attend to important stimuli and ignore irrelevant background noise.
Is there a specific system for pain modulation?
Yes, a very important and medically relevant one, the endogenous analgesia system. This system is specialized to suppress pain, particularly under conditions of stress or danger.
Like the examples you gave earlier, soldiers, athletes not feeling pain immediately.
Exactly. That's not just adrenaline or shock in a vague sense. It's this specific neurobiological system kicking in. It allows focus on survival or performance by temporarily inhibiting pain signals.
Which brain areas are involved?
Key centers include the midbrain periaqueductal gray, PAGE, the locus coeruleus in the pons, source of norepinephrine, and certain medullary raphe nuclei, source of serotonin.
And how do they suppress pain?
Stimulation in these areas sends signals down the spinal cord and brainstem, where they inhibit the activity of nociceptive neurons, including those crucial second-order neurons in the dorsal horn and trigeminal nucleus that give rise to the spinothalamic and trigeminothalamic tracts. They effectively turn down the volume on the ascending pain signal. Other descending inhibitory pathways also originate in the sensoromotor cortex, hypothalamus, and reticular formation.
Does this system use opioids?
A major component does, yes. It utilizes the body's own endogenous opioid peptides, things like enkephalins, dynorphins, and bodendorphin. These peptides act on opiate receptors located on neurons within the pain pathways.
So morphine works by hijacking this natural system.
Essentially, yes. Morphine and other opiate drugs activate this same system. They often work by inhibiting inhibitory interneurons in places like the PEG and medulla. By inhibiting the inhibitors, they disinhibit the descending pain control pathways, leading to stronger suppression of pain signals downstream.
How do the opioids inhibit the pain signals directly?
Enkephalin, for example, can act both presynaptically on the terminals of primary nociceptive fibers, reducing their release of transmitters like substance P, and it can act postsynaptically, directly inhibiting the second-order projection neurons in the dorsal horn.
Can this opioid effect be blocked?
Yes, by opioid antagonist drugs like naloxone. Giving naloxone can prevent or reverse analgesia caused by morphine or endogenous opioids. This makes naloxone a really useful tool for determining if a particular analgesic effect relies on the opioid system.
Is all endogenous analgesia opioid-based?
No, definitely not. Some descending pain control pathways are unaffected by naloxone. For example, serotonin released from those raphe nuclei neurons directly inhibits nociceptive processing neurons in the dorsal horn. And norepinephrine released from locus coeruleus projections and other brainstem neurons also powerfully inhibits nociceptive neurons, partly through interactions with the opioid system, but also through non-opioid mechanisms. It's a complex interplay of multiple neurotransmitters and pathways, all working to allow the brain to control its own perception of pain.
Okay, let's try and unpack all of that. We've really taken a deep dive into the somatosensory system today. It's incredible.
It really is.
From the amazing detail of individual receptors sensing the slightest touch or temperature change, to the complex pathways racing signals up the spinal cord.
Crossing the midline at specific points.
Right, and arriving at those specialized processing centers in the thalamus and cortex with those detailed body maps.
We explored how your body differentiates, a gentle caress from a sharp pain, distinct pathways, distinct receptors.
And how the brain isn't just passive, it can actively turn down the volume on sensations when needed through those descending pathways.
What's truly profound, I think, is how this incredibly intricate network, specialized cells, unique proteins like PaSO2 and the TRPs, precise anatomical pathways, allows for such rich, specific, and diverse experiences of the world and our own bodies.
Yeah. From the properties of FA and SA mechanoreceptors letting someone read Braille,
to the specific TRP channels like TRPV1 or TRPM8 alerting you to dangerous heat or cold. Every single sensation is just this symphony of biological precision.
So, thinking about this for the listener, what does mastering this level of detail mean for you?
Well, the next time you feel anything, a pinprick, a comforting hug, even a deep ache, maybe pause and consider that silent, tireless dance happening inside.
The piezo2 proteins responding to pressure, the TRP channels firing off for temperature.
The intricate filtering happening in your dorsal horn, your brainstem, your thalamus. How might really understanding your somatosensory system at this kind of depth change the way you perceive everyday sensations?
Or even how you think about complex and challenging conditions like chronic pain or neuropathic pain. It moves it from a vague feeling to a complex biological process we're starting to truly understand at the molecular level. Something to really mull over.